Bismillahirrahmanirrahim. Assalamu alaikum, dear students. My name is Zakirullah, and I am a lecturer, Medical Laboratory Technology, Institute of Biological Sciences, Sarhad University of Science and Information Technology, Peshawar. Today, our first lecture of online classes will be regarding the reception and collection of biopsy specimens, that how different types of biopsy specimens they are received, they are collected in the laboratory, and how they are processed. We will discuss the processing and the other procedure regarding biopsy specimen in the next lectures. So starting from the beginning, sample collection. How a biopsy specimen or sample is collected. The procedure is that when any surgeon or doctor remove a specimen or an organ or a tissue from that tissue is called biopsy, because that sample is taken from the living organism. So to preserve this tissue, to preserve this organ, this it is placed in a compound, in a chemical substance which is called fixative. Fixative are those substances which preserve the structure of the cells and the structure of the tissues. Routinely, we use 10% formal saline fixative, which is a very good and which have a good advantages as compared to other fixatives, which also have certain advantages. But formal saline, it is one of the best and one of the routinely used fixative for the biopsy specimen. The fixative should be at least 10 times of its volume more than the tissue. For example, if a tissue is about 10 ml or about 100 ml, then the fixative should be 10 times more. In case of small biopsy specimen, in a small container, the whole tissue or the whole biopsy should be at least immersed in the fixative, so that it should be properly fixed and then processed properly. Containers for specimen. Bottles or jars with screw tops of suitable capacity are required for collection of biopsy specimen, because when we put fixative and biopsy in that container, to make sure that it does not get leaked or the contents of the specimen they are not lost, so they need to be screw topped. For large specimen, for example the whole kidney or a limb or any other specimen, there should be a large container of suitable size. If the container is small and tissue is large, then the tissue should not be forced or squeezed into the container, because if we forcefully insert a tissue inside a container, then it can cause problem in fixation, and the fixation may become inadequate. And if there is improper or problem in fixation, then what will happen? It will cause autolysis. What is autolysis? Autolysis is a process in which a tissue degenerates. When a tissue is removed from the body, all its processes are stopped and the destruction process of the tissue starts. This destruction process of the tissue is called autolysis. This autolysis it becomes difficult for a person to judge any disturbance, any pathology in the tissue when the tissue become autolyzed. So we need to properly fix the tissue, and all the structure should be properly preserved with the fixative. So for this purpose, we need a specific and suitable size of container. These are the different sizes of containers. We can see small containers for small tissue and other a bucket size of container for large size of tissue. So the lab should have a suitable and every size of container, because we need to have all these, as sometimes the tissue is larger, so we need a larger bucket, a larger size of container for large organs. Large specimen should be brought as such to the laboratory in a bucket or other suitable container, because if we do not have a large bucket or a large container, if a specimen is too large, then the largest size of container, if we have a container but the specimen is larger than the largest size of container, then make sure to bring the tissue, to bring the organ directly into the lab in a bucket or any size of container without fixative. For amputated limbs, they should be wrapped in a rubber sheet and then it should be brought to the laboratory. Because we do not have specific container for limbs, therefore it should be brought as such into the laboratory, and it should be wrapped in a rubber sheet, just like in the old days. A rubber a type bucket were used for water, means like large tumors or spleen or liver. They should be cut into different slices and then placed in a container full of fixative, because the fixative will fix all the area, the outer side and the inner side of the tissue or the specimen, so we can process and then stain and then microscopically check all the areas of the specimen. So therefore, for proper fixation, the large size specimen or solid specimens, they should be cut into different slices. See a large size of tissue on the left, which is squeezed into a container. We can see that the tissue structure is deformed, it is disturbed, which can create problems. On the right side, there is another tissue, a liver tissue, and it is cut into a specific slices. So for large tissue, as we already discussed, the tissue should be sliced into different sizes and then make sure to fix it properly and then transfer it to the lab for processing. For hollow organs like stomach or small intestine, they should be open at both ends, or they should be cut along the whole path, so that the inner side and the outer side both are fixed, and we can take samples from the inner side as well as the outer side. If jars of fixative are not available, specimens should be taken freshly to the laboratory or wrapped in a moist cotton wool and put in the refrigerator overnight and then processed. Sometimes the organ or the tissue or the specimen, it is very large, so we do not have a specific jar, a specific container for the tissue. Therefore, we need to bring that tissue directly to the lab and make sure that it should be covered in a cotton, and that cotton should be soaked in a fixative. One thing we should do, that we should never ever put a specimen in a simple water or a normal saline, because in simple water or in normal saline, It provides a good environment for the bacteria or for the microorganism which causes degeneration and autolysis of the specimen. So therefore, we should never put any organ or any tissue in a water or normal saline which is diagnosed or which are transferred to the histopathology laboratory for histopathology report. Rapid frozen section is another type of procedure which is an emergency procedure. It is done in a machine called cryostat, in which the environment is about minus 25 degrees centigrade or minus 13 Fahrenheit. For rapid frozen section, the lab staff should be informed at least one day before the procedure or 1 hour as early as possible. The lab staff should be informed, because when lab staff is informed, they will prepare the machine for the procedure, because the machine need to be prepared early, and all the instrument inside the machine, to get them ready, they need a specific time. And rapid frozen section, we know that it is an emergency condition, so the report is prepared in emergency situation. Therefore, the surgeon need to inform the lab staff before the procedure. For rapid frozen section, the specimen should be brought unfixed to the laboratory without delay. If a specimen is removed during a surgical operation or a surgical procedure, then that specimen should be brought to the lab without adding any fixative to that specimen, that tissue, and then in the lab it is processed in the cryostat or cryotome. Then slides are prepared and stand, and then a pathologist make a report. Then when it is suitable for a doctor or a surgeon, then he can go further as per the investigations of the lab. If a doctor thoughts that there is no need for rapid frozen section, he should inform the lab staff at time, so that they should carry on their routine work, because rapid frozen section is an emergency procedure. Therefore, the doctor should inform the lab staff that he postponed or cancelled the rapid frozen procedure. Therefore, lab staff should be informed timely. Biopsy specimen which are transported to the histopathology laboratory, they should have a request form. This request form should contain all the related information regarding the patient, regarding the sample, and also ward, hospital, and other related information which should be mentioned in the request form. A single request form is enough for a single disease. For example, if more than one biopsy is taken from the same patient at one time, a single request form is enough for all specimens for to date single pathological lesion. When there are two or more unrelated pathological or there are two or more unrelated diseases or different diseases, separate forms should be filled with each specimen, and that specimen should be mentioned on the request form. If there is any previous samples or any previous reports from the same patient, then this should be also stated in the form along with the previous ID and their date and their reports. And in case of if the investigations are done in another laboratory, then it should be also stated in the request form that the investigations are performed in the other laboratory than this one. All this information should be filled, and all the request form should be filled by the medical officer or the in charge of the case, because it is the duty of that medical officer to make sure that all the required information are written in the request form. Apart from the usual particulars required on any request form, just like the age, the gender, the address, ward, bed number, and the clinical history, the data must also have any specific information regarding to the present illness. If there is any special sign, symptoms, or any specific information, it should also be written in the request form. The request form in surgical condition should have the additional information. One is the nature of the operation performed, that what kind of operation is performed, either it is mastectomy, partial mastectomy, or full mastectomy, or it is should also be mentioned that either the whole the organ or whole the system of the patient is removed and it is sent to the lab, or only a part of the system or a part of the organ it is removed from the body and sent to the lab. So both these information are must in case of surgical procedures. They should be attached, they should be sent to the lab along with the other information. Labeling of the specimen. Labeling of the specimen is also very important for the histopathology specimen or all the specimen which are sent to the lab. The specimen should be correctly labeled by the medical officer, including the name of the patient, the ward, the hospital, and the specimen type, along with the date and time of the obtaining the specimen. Because if we mention all these things, then when a pathologist is reporting such kind of biopsy specimen, that he should also mention such things. These particulars should tally with those stated in the associated request form. That all the information written on the specimen and all the information written in the request form, they should be same. If there is a difference in both the information, that the sample should be rejected and it should be sent back to the hospital, or the medical officer should be informed to make proper corrections. The different types of specimens, they are sent to the laboratory, histopathology laboratory, for for biopsy of the kidney specimens. They should be collected in 10% formal saline and sent to the lab. The request form should have all the clinical information along with the lab investigations, which include the urea, creatinine, and all the clearance, 24 hours urinary creatinine clearance, or along with the other sodium, potassium chlorides, and all the minerals that should be also tested and sent to the laboratory. If renal specimen or renal biopsy is sent to the lab for immunofluorescence, then it should not be sent to the lab in the formal saline. Rather, it should be sent to the lab in a normal saline, because normal saline do not disturb the immunofluorescence and the immunofluorescence staining. Therefore, if a renal biopsy is investigated for any immunofluorescence, then it should be sent to the lab in the normal saline. Liver biopsy, it should be also sent to the lab in 10% formal saline, and all the related lab investigation should perform, including the liver functions test, all the enzymes and bilirubin, albumin, all the proteins that should be checked. And if there is hepatitis, then also hepatitis markers should also be performed, and all the related information regarding the liver function test and the hepatitis markers, they should be written in the request form. If liver biopsy, it is tested for storage disorders, then it should not be preserved in 10% formal saline. Otherwise, all the structure will be distorted and disturbed. So, for storage disorders, liver biopsy, it should be collected in absolute alcohol, 100% alcohol. Therefore, it should be noted that for liver normal routine histopathological investigation, liver biopsy is collected in 10% formalin, but for storage disorders, it should be collected in pure alcohol. The bone specimens, it should be also collected in 10% formal saline. The information which is follow with the specimen should be the age of the patient, the gender, the biopsy site, and the clinical history of the patient. Along with the X-ray or other radiological investigations, which it should also follow the specimen. The radiologist's opinion is must regarding the bone specimen. So that was the lecture regarding the reception and collection of specimens. I hope that you have learned that how to receive a biopsy specimen and what are the fixatives which are routinely used for collection and preservation of biopsy specimen. The other things we learned that we need a suitable container for biopsy specimen along with a request form, and this request form should have all the relevant information and specific investigations. Like in case of renal biopsy, we discussed that it should contain the renal function test, urea creatinine, and in case of liver biopsy, it should also have the hepatitis markers and the liver function test, which include enzymes, the proteins, and the bilirubin functions and their quantity in the blood. So that was our lecture for today. Hope that you learned most of the things. We will be looking in the next lectures for the cytopathological specimen and their collection. Thanks for viewing the lecture. Your suggestion and your question, they are welcomed. Thank you. Assalamu alaikum.